medical image processing. Hi, I am Konstantin Bychenkov. I'm head of Aligned Research Group. We deal with computer vision, with augmented reality, and this, of course, is about image processing. And in the last 2 years, we are involved in a huge medical project, which started new life in our company. In my presentation, although it's about medical imaging, we shall not see many medical images. We're going to discuss the main approaches, the main tasks of the company. On the one hand, we would like to check ourselves, as Niels Bohr said, if you are a physicist and if you cannot explain your problems to a 5-year-old child, then something is wrong with your research. Same here, if you cannot explain, so you are doing something wrong. Maybe you will find something interesting here as well. Now, well, actually, big data in medicine is developing very fast. These are the main areas. You can see them in the slide. We are going to talk about research and images, something which is closer to my company. Although we have a lot of data in the world, and I cannot but tell you about the genomic. Last year, the Institute for Systems Biology has received a grant of the National Cancer Institute, or National Institute of Health, which is in charge of healthcare grants in the US. They, together with Google, are going to build a cloud system for genome processing. It's cool, and Google is on the one hand. It entered medicine from a different door. They wanted to develop a healthcare platform for everyone to place the data there. No one wanted to do that. Everyone laughed at them. But through that institute, they will aggregate medical data. It's a cool subject because cancer treatment gives you a lot of field for research. Big data is needed because cancer is variative. There are a lot of versions of the disease, and development of the cancer cell also changes its gene code in the process of its development, and the usual approaches cannot be applied. From time to time you see sensational claims that another treatment for cancer has been found. Yes, it works, but at a certain stage only, and it doesn't work on other stages. The more data we have, the more options we have, the more opportunities we shall have for the development of personal medicine. Everyone believes that the more data we have, the faster we're going to implement personal medicine, and we shall treat a person with his or her personal genetic code. In general, the data comes from different sources, and these are the sources. It's X-rays and CT scans, MRT scans, which is a must-have for any clinic. All the clinics abroad have such systems and data storage sites. We have a federal program in this country as well, a federal image archive. But even abroad, a lot of clinics have been deleting such images.
And so they use our data banks. And so last year, so London College reported that they have their own cloud CT and MRI. So then the photos, the image archives, they grow by 20-40% every month or every year. And so all together, so this data, images, genome, pictures, etc. So they open up possibilities for personal medicine. So therapy, customized, pinpointed, client-centric therapy. And so when they create big databases, anonymous, large-scale collection images, and we and then the precision is on the rise. The diagnostics devices, so CT and MRI, is getting more precise, and we can we certainly then CT and MRI that now they are so they are interpreted by diagnostician. But now they're going to depart that approach, and there will be then there will be more objective analysis will be done in a protocol. So to make it more objective, to make it more thorough, to make it more uh, 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 and so then say so to make it accurate, to make it objective, so just we're just waiting for another professional who would just come and and make the final diagnosis, and because we don't have many good diagnosticians, and that is why so this ability to make the right diagnosis the first time will help a lot, because our healthcare system deals with other issues. Europe and the United States, so they have accumulated a great deal of data. CDAS, this is the US database. It's not available. You have to register to get. You cannot get this information from Russia. But since we are dealing with CT and MRI, and so it will be great to grab it, to get your hands on this, because construction algorithms, statistics, screening attempts, reconstruction, and so different algorithms of the quality and accuracy of CT screening from different manufacturers, from different vendors, and we can see who works well, who doesn't work well, and that's lots of additional information. So this you can get an access. This is a functional MRI. Yes, it's a wonderful thing because it does the anatomy, and then it reads the biological activity, and thus you can, for example, so the activity of the brain, so the way the brain functions, so you can just get access to it and work on it, and it's cool. And neuroscience is burgeoning, and so we'll talk more about MRI. Then this military neuroscience is developing very rapidly. So any big country, powerful country, so that it invests a lot in its military. So they certainly developing this war interface, war brain interface. So very soon we'll learn about human brain much more than we've learned so far, and so some special agencies will know even more about the way the human brain functions. So what can we do with functional MRI? So we stimulate the brain, and then we analyze activity and behavior, and so then there's this statistics, some figures. So we analyze the activity of the brain of a fish. It's easy. So it's 100,000 neurons and 1 terabyte of data. That's it. But if it's a mouse, so then so we can only so how much data storage is required. But the human brain, so in 1 pixel is 50,000 neurons. That's why the human brain's behavior can be evaluated very tentatively. And so then there's a big problem of big data. Big data issue is very high on the agenda, and because it's so it won't fit into any server. It won't fit into any. So we need to distribute this huge raw data, big data, taken which records only one human man's brain. They will need to be distributed, shared, and visualized, explored, analyzed, and it's much more difficult. So now just let's move over to neuroscience, the typical questions. So then we try to understand how the nervous system functions. So this during this MRI, during the different stimuli. So then the brain acts differently, and then the data is processed. So the simple method, the most simple method that can be applied, is the PCA method. PCA method to reduce dimension and eliminate patterns in data. And because it's big data, so we use Spark, for example. And so Spark. I think that you know about Spark. I don't want to go into detail, but we what we do, we love this system. It's great, because jointly with Python, so that can be attached. So very powerful math libraries of Python and plus Spark, which deals with data very well. It handles data. And that's puts on the cluster of the distributed system. So then the SPARQL, so which was inherited from Hiva. From Java, and so then the streaming is wonderful as well. So we don't need so much that streaming property, but for if you say selling DNA servers, or if you are a cell phone operator, this cluster function is very good. Then there is machine learning there as well, and then they have Scala, Java, Scala, Python. They have it all. So if you want just to develop a new program, so you can select Scala. If you're a researcher, that's very good. Python's IP. So then it's a wealth of information. So the core is responsible for task management, memory management, for robustness, sustainability, and because if certain things works slowly, or some work faster, so then you can just concentrate on them and so on. And so, for example, data source management, API for data collections, RDD. So on Hadoop, you cannot move so fast as on Spark. Spark, because Hadoop works on the disk array systems, and Spark is quite different, much faster, resilient data, and then data collections. So that's out of the box. So simultaneous access to data collections. Very powerful instrument. And a typical Spark program looks like this chart. So there is a driver that launches blockers, and each one has a certain task. So it's all configured from the very beginning, and it's quite transparent, and can be superimposed over Amazon, and it works well. So all the data sources are shared between workers. Also, so then that's about Spark. So PCA. I want to give us an example of the information that's why we start with fish. Fish is easy to read. So we have a set of pictures about the brain's activity of that little fish, and so I want to analyze the brain activity of this little fish, so without any losses in info. So what center is responsible for what, and for this we'll do the method of main components. Specifics of this method is we are attempting to reduce the size of data. So 240 seconds to squeeze it into 1 second, or 2-3 seconds, and see how, and without any losses, to see the picture in time, stretched in time. So 2-3 snapshots, and for the purpose of the PCA formulation. So the initial data that we collected. So we found this matrix, convolution matrix, identify main components. I don't want to talk about that, though I have slides. And the way it works is as follows. In Spark, it's quite straightforward and very easy to represent. So we download so the intensity of pixels of the picture of the brain. Then normalize, streamline, then apply. So then bring it down to 3 components, and then take the first 2. And using this method as the main approach, the first 2 components are sufficient to get the main picture, and the main picture will look like this: the first component, second component, So these two gray photos are very difficult to understand what they mean. But if you put them together and you can then you convert and align them along the intensity of brightness, and then you can see on this colored picture already what you see. So it's color coded. So all 240 seconds so we squeezed into 2 seconds, and depending on the stimuli, so you can see what stimuli and what centers of brain activated. And so there is no one single source in the brain that can process these stimuli, because they are located in the brain in different places, and there's no single point. And it applies. The same is true of men. And so this is something that neuroscience uses a lot. Then next, so you can take it one step further, and we are in the middle of a good project with Lasagna University, which you don't have to do this constant processing of the data that fMRI gives you, because there's lots of information. It's quite troublesome. But having collected certain statistics, you can move over to a different level, to building models already, models on the basis of the material. So the model of the brain activity, and then taking it from there to calculate what response would depend on what kind of stimuli. And I cannot talk more about that because we just started. And but the main approach is building a dynamic causal models. It's only one slide, causal models. Then another thing that we use a lot. So there is another challenge. So then to make it an objective, to make research more objective. So CT and the MRI. And this is an outstanding issue globally, because when especially oncology is treated, so the oncology sometimes may be very complex, and so in the picture it's very important to identify the points of oncology, so the foci of oncology, because the internal body looks like a plastic bag full of oranges, and even if it's the last stage of oncology, so you have to fight the disease until the end, and nevertheless, and to identify all the points of disease is impossible to do it manually, and so then this automatic finding of those foci of inflammation of oncology sources. So then scripta choralica, whereby they're attempting to this multi-layered space to introduce a certain standard, and using this yardstick to identify the intensity. And so by tuning the intensity and using the descriptors, so you can identify the areas of oncology, of pathological changes. Yes, so then to do this comparison, this neuron network, neuron mesh, as was identified in the 80s. So here, from the low level, they take features, there's low level features. But as we go more, just looking for new features, so the descriptor choralica does that. So then the classifier works, and you can find certain comparisons, and it's like a puzzle. So the same choralics, and so if you break them down into samples, then this works very well. So then good mesh works well. So that develops algorithms on the basis of choralicas, but manually. So there was a good presentation report as automatic identification of emotions. So choralics 1, 97% authenticity to 96%. But the neural networks a new thing, although it appeared 20 years ago. But the typical application, for example, so then they detect so the faces, for example, very well. Um, so selecting to configure the this network. So it's a famous picture. So then you can see the guy who is invisible is sitting in the tree. In the tree. Uh, 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 hmm. So we want this instrument from the viewpoint of segmentation. Segmentation. So where we use it. We every MRI cross section, we need to put. So we need to identify that this is pathology. This is pathology. To identify all the foci. Yeah, and well, I want to show you because the idea is just to identify on MRI all the pathogenic changes, dysfunctions. This slide is very important. Then, so then the spatial correlations don't work by themselves because the local minimum problem is there. And so then on top you can see this photograph. This red dot on the person's face, and the elbow is so then the system, the mesh, thinks that elbow is the same as the nose, as the nose. So then it's a probable analysis of the pictures. So then there is a white pixel, then white 6 pixel, and then red pixel. Then probably it's a traffic sign. If it's red, black, right, and then blue, then probably it's not a traffic sign. And so this method of exclusion. So we are training this system, and so that's quite accurate. This, for example, this red spot covering the face says that it's a face. Mhm. Yes, so we use Café. Café is a wonderful instrument, good for the researchers. This is mainstream now. And so anyone who is involved in research, they do it on Café. Café is wonderful. Main advantage that they manage to do very good language with networks configuration and training strategy. You don't have to program this networks configuration. You need to develop. So then it supports GPU from the box. GPU. This is good because these approaches were not applied because no one wanted to wait for a few days. Then Café is developing very rapidly, like any other instrument develops fast, but it has a negative side. If you want to move from research to production, then there'll be difficulty. Because if you want to put Café on Windows, so if you're in production, it may be needed, and so we try it, and it's not worth it. Because to put G logs, to get G flags for Windows, it's a torture. It's very difficult. It works, but not very well. Yeah, there is no backward compatibility, and so they train differently. Learn differently, and the configuration so described in the older version, so you will not be able to inscribe it into the younger version. And the cafe. So then another thing I wanted to show you, some little footage how it works. How it works. Yeah, so then we did it jointly with the Samara University. I'll show you how the system with the reference networks, how the machine segments the pathology. This is liver. So how it will be configured and segmented and identified. So the beauty of the system, it works. So you put a mask over overlay it over the liver, and you press the button, and it starts looking for the areas of oncological de novo formations. Yeah, so green is this is our network in progress, and these are the results. Let's compare, and so we'll get it from the researchers now. So. So this is what the doctor identified. So the challenge is that the oncological diffusion from good tissue to bad tissue is high. So that's why to delineate it, to set a boundary. So all this successful research tells about metrics. So volume, if it coincides 20% volume-wise, it's a success story already. So if 20% volume uh, uh, coincides, it's good. Sometimes it's a false trigger, and I have to admit that. So this is long way from production stage. So 95% of research so will definitely break this algorithm, and so that's why it needs to be trained. And so we need a lot of data because this reference network requires a lot of data.
This will eliminate the problem of configuration. If we have a different kind of a disease, polycystosis, for instance, when we have a lot of wrong cells. I don't know how to work in that case. In that case, our system doesn't work. So we try to do something about it. Everyone hopes that within the next 5 years, segmentation will work and it will help us all. And we are also in that race. Thank you. I presented to you questions, please. Thank you very much. You mentioned Spark and Cafe. Spark, well, they do not related to any together. So there are different groups, different teams. They they work, they develop. Spark is a bit different, and they work with each new product differently. They have a database, they have ground truth, they are expanding ground truth. Spark is different. We have a partner, we work together. Spark is deployed there at the partner. Our data scientists work at the partner, and that's all. Why do we use it? What do we use it for? We process big data, not separate images. We process big data. If we take, for instance, human brain, we have a lot of images. Of course, we compress them. And also, when the images are not processed any longer, when we have a model, and now, well, how to say, we try to prove our model in practical situations by practical experience. We know that this data must provide this kind of answer, and we see whether it gives that answer or not. We have a lot of data to download. A lot? How much? About 50 terabytes. Not too much, but quite a lot. What is hierarchy? Hierarchs method. Well, hierarchs descriptor. Well, it's an attempt to to use that method in multi-dimensional space that would help to position each point in this space in comparison with other points. Then, by knowing the intensity, by comparing it to the ground truth, we can more or less precisely, well, Haralick's descriptor helps us to position the point. Knowing the range, knowing the point, we can build a mask and it will look like a stellar sky with some constellations, and then we can do with that constellation anything we like. We can use clustering. If this is a tumor, a neoplasm, then we can clusterize it, putting some kind of boundary around it. If we want to select some section of a human body, and if we know the topology, we can have, for example, an average human liver, the way it should be, and see the way it looks now in comparison to average. It gives a lot of interesting data. Thank you, thank you.